warahmatullahi wabarakatuh, and a very good morning. Thank you for joining us today for one of the GSM technical talk for 2024. So under the Quaternary and Marine Geology Working Group, we have invited Dr. Fatin Izzati Minhat, Associate Professor Dr. Fatin Izzati Minhat. Here we have on screen to give some talks on modern foraminifera and their application. And one of the reason is actually to share and also to expose and perhaps to encourage more people to know about other types of research that has been done, especially on marine geoscience. So both of us are from Universiti Malaysia Terengganu under the program of Marine Geoscience, and we are relatively young. We only have our first cohort graduated, so I guess this is also an effort to sort of like encourage and also expose that there are other field that is quite involved already for quite some time here in Malaysia. Okay, so before Dr. Fatin starts her talk, I would like to introduce her. So I asked her to send me a very short biography about herself. And the first thing she said that she's born in Penang and a true Penangite. So I guess I have to say that because that's what she said. So she started her higher degree education in Universiti Science Malaysia and obtained her BSc in Applied Biology in 2010. And then that's where she got to know about foraminifera. And in her own words, she discovered that biologists somehow do not favor foram. So we have to ask her what's that about. And myself as a geologist, I've known about since BSc, we've learned about foraminifera. And then she delved into the world of earth science, which is specifically on marine science during her MSc, also in Universiti Science Malaysia. And she then did her PhD in Universiti Malaysia Terengganu, whereby that is when she actually did work on foraminifera, which focuses on reconstructing sea level during Holocene. So I guess without further ado, I would like to invite Dr. Fatin to then maybe talk more about foraminifera and introduce us to the world of modern foraminifera. Okay, the floor is yours.
Thank you so much, Dr. Khaira. Thank you for having me here today, for the opportunity, the technical team of GSM as well. So today I am going to share with you a little bit on forams. If you are from, geoscience or geological related field, I guess most of you are very much familiar with forams. So let's see. All right, so today a little bit on modern foram instead of fossil forams. I would love to talk about the application because you see in today's ocean, you still can find foraminifera. They've been there for so long throughout history. So we are going to talk about the one that is still here. All right. So just an overview of today's talk. I would introduce or reintroduce what is foraminifera, talk a little bit about where do they live, what do they eat, how do they reproduce, and some sort of like what are the different shell types or test type, and a little bit on the evolution, and then I will touch a little bit on the application of foraminifera. All right, so I think Dr. Khaira has covered most of it. I am currently teaching in UMT, teaching marine geoscience. So usually the courses that I teach is much more related to biology, such as paleontology or geobiology, things like that. Because I'm still a biologist at heart, as Dr. Khaira mentioned previously, I like biology. I mean, when I was accepted to boarding school, I was once in Science 3.3. if you've been to boarding school, you know, we tend to be very sleepy in every class, but I rarely get very sleepy during my biological class. So right then, during my high school years, I know that, you know, I wanted to be a biologist. I not necessarily wanted to be a doctor, but then I wanted to be, you know, a biologist. So when I started my higher degree course or BSc in USM, I chose to do applied biology. And then we can choose what is our major during our second year. So instead of choosing microbiology, which is much more related to Forem, I chose aquatic biology because to me, the marine world is such a mystery. And of course, I like to do the field work, and microbiologists seldom get to go outside. So I graduated as a aquatic biologist, and then I started to do my master science. All right. So what happened is that during my master study, I am supposed to carry out a research on myofauna. So I'm not sure if all of you are very familiar with the terms myofauna, but basically myofauna is not as microscopic as it is, but it's not as large as the megafauna. So it's like in between. So I was trying to identify this microfauna, but somehow I see a lot of these strange looking animals or organism that looks like a flower to me. So I do not even get any myofauna during that time. So I asked around, and most of our science officer do not have any idea what is it. Even my supervisor was not even aware what is this beautiful little flower in the sea is. So until I met the late Encik Omar, so he said that it could be a foraminifera. So that is how biologists we don't really know what foram is. We don't learn about foram. Somehow they just, you know, take out foram from our course or teaching lesson. So I discovered foraminifera accidentally. But then I still carry on doing foraminifera during my master degree, which mostly trying to use foram as pollution indicator. So what we did was we sampled the forams, we look at the water quality, we look at the sediment quality in the area, and we try to see whether, you know, can foraminifera become an indicator in that area. And then when I wanted to do my PhD, I realized that, you know, in the biological world, I don't have that much support in forums. So I started to reach out. I was like reaching out internationally. I was reaching out internally, locally. And then I found UMT. So during that time, UMT was, one of the professor is having a few students that is currently doing lots of foraminifera work, including Dr. Zalia, if you're familiar with her work. So I reached out to UMT, and then UMT somehow connected me with most of these foraminifera expert in Petronas. So that is when I discovered, oh, somehow foraminifera is much more related to geology rather than biology. This is why we don't have forams in our biological work, biological book or things like that. So I continue my PhD, but then during that time, I was looking from the perspective of geoscience. All right, so enough about me. Let us see what is foraminifera. Okay, so foram is actually a single cell organism. We as biologists, we like to call them protists. So they are very much like our cell, but they are only single cells organism, which means one cytoplasm, they can have a lot of nuclei, you call it. they are very, very similar to amoeba. If you remember our high school teaching, amoeba, they are very much similar to amoeba. But the amazing thing about foram is that they have this shell-like structure that protect them. So this shell-like structure, instead of calling them shell, paleontologists or micropaleontologists somehow term them as tests. So we don't call it shell, we call it tests. All right. So if you know foram, it has been very famous in fossil records. They have been there since the Cambrian. So if you were to look back in Cambrian samples, most of these foram can either be naked foram, means they don't have tests yet, or they may have a specific test that is made up of sand particles. All right. So from there on, they will evolve to the foraminifera that you look here today. This is calcareous foraminifera, which means they are made out of calcium carbonate. All right. If you wanted to see foram, you need a sample from marine environment. So what if you have a fluvial sample? No, you won't see any foraminifera unless it has been transported, or if that area has somehow has a little bit of salinity, then you might see foraminifera. But if you go further up on the stream or river, foraminifera will just disappear. So they are very good indicator for marine environment, which means they are there starting from the estuary area, they are there on the shelf area, they are there in the slopes, even in the deep sea area. And mind you that they have survived all, not just one, all mass extinction events. Especially the agglutinated, or what geologists usually call the arenaceous foraminifera. They've survived every mass extinction event. All right, so what is the average size of foraminifera? Well, if you don't like microscope, it's going to be very hard for you. So initially, when I started off my research during my master, I really do not want to do anything related to microscope. But then when I found foraminifera, I somehow, I fall in love with forams and I can't get enough of them. Even right now, if we are like very busy with our, teaching works, administrative work, I still have to look foram under the microscope. I got addicted to it. Well, I think micropaleontologists can relate with me. We enjoy looking at forams under the microscope. They come in various shapes. You can see a lot of them with different big size. This looks like a flower to me. This is what I call like a flower, but this is a planktonic foram. They have a very roundish shape forams. They have elongated foram. You have a very oval and flat foram. So they have various shapes. And mind you, there is like around 40,000 species right now, fossils and modern foraminifera records, and a little bit about 3,000 to 4,000 species of planktonic foram. All right, so majority of the foraminifera is calcareous species. As I said, they evolved through time. They started from the agglutinated or arenaceous. Arenaceous means they built their shell by collecting sediment surrounding them, and they will glue this sediment together to make their own shell. So those are the more primitive forams. But as time goes by, they started to evolve, and somehow foraminifera has been able to, make their own test or make their own shell, which is calcareous. So before, the calcareous, you have like in between try and error process, as you know, evolution goes that way. But right now, you can see a lot of modern foraminifera belongs to the calcareous group. Okay, so where do they live? As I say, they are exclusively marine, you can see here, this is a very fine diagram on how different shape of forams prefer different places in the marine environment. So if you are looking at shelf environment, you usually get this kind of elongated foraminifera, which is much more agglutinated foram. They are very close to the coastline, and as you move towards the outer part of the shelf, you started to see the dominant species of foraminifera started to change the shape. And if you go further down, the shape still change. And if it's much more deeper than 3,000 meter, you will not get calcareous foram because of the carbonate composition depth. So you will started to see the agglutinated foram again. So if you have deep sea specimen, you will get a lot of agglutinated foraminifera. All right. So, but one thing about foram, agglutinated foram is that they seldom get preserved. They seldom get a good preservation. So it's very hard to see them in very old samples. Rarely you get to see them in very old samples. All right, so other than the benthic foram, so we have two types of forams. So this is those that stays on the sea floors, but we also have another types of forams that floats. So this is what we call the planktonic foram. So the planktonic foraminifera is all calcareous foraminifera, right? So they are the modern one. You started to see planktonic foram during the Mesozoic. Prior to that, you only see lots of benthic forams. All right, so starting Mesozoic, you started to see a lot of planktonic foram. They started to take, they started to float, and a lot of different shape of planktonic foram by then. So where do the planktonic foram live? Amazingly, even in the water column, foraminifera somehow has their own zones. We call it the zonations. Some forams, they like to float very close to the surface. Some of the foram are very close to the thermocline layer. Thermocline is where you get a sharp decrease of ocean temperature, so we call it the thermocline layer. But we rarely get foraminifera in the deep water column because there's no more sunlight by then. All right, so they need sunlight for them to be able to float here, and food. The food availability is somehow on the surface of the ocean and the thermocline. So you can see how the shape changes throughout the water column. All right. So this is what we are here for, the application. Because since most of you has a little knowledge on foraminifera, we also know that in geology or geoscience, foraminifera has been used exclusively in oil and gas exploration. So in 1920, this is the book that has been published in 1920. It's a very famous book, Cushman. They have a foundation named after it. It's called Cushman Foundation. They focus on foraminifera study and other related micropaleontological study. So basically, what happened in the 1920 is that the book is being published and they highlight the economic use of foraminifera. Why economic use? So the reason why it has such title is because back then, foram is being used by biostratigrapher to determine the age of the well. So when you're doing oil and gas exploration, you need to know the age of your well. So this is where micropaleontologists come in. And this is why the name or the title of the book itself is being related to the economic use. So if you are very familiar with foram in biostratigraphy, we usually use, especially planktonic foraminifera, because they have a great first appearance datum, the FAD, and they also have this LAD. So basically, planktonic foraminifera only live at, certain species live at certain time of the year, time of geological timeline, and then they disappeared, and then a new species come in, things like that. So because they're short appearance, somehow this planktonic forum is good to pinpoint what is the age of the well. So that is why we use planktonic forum. Why not use bigger fossil? Because the abundance of planktonic forum, if you look at marine sediment, you can see sometimes up to 80% of the sediment is made up of planktonic foraminifera. This is how the foraminifera is being used as biostratigraphy. All right, so what else? So since we are talking about the biostratigraphy tool, if you look here, this is a picture of a KT boundary. So if you are much younger, we change it to KPG boundary, Cretaceous Paleogene boundary. This is here in Gubbio, Italy. So what happened is that other than biostratigraphy tools, Forum is also good at, showing what happened between the two timeline, these boundaries. So what happened is that if you look here, you get to see the Cretaceous sample here, and then a very small area of mud in between, and then you enter the Paleogene, which is the Cenozoic. So what happened is that some micropaleontologists has sampled the sediment between or rocks in between, and they extract foraminifera from these two different boundaries. And what they get is that if you look at the Cretaceous period is a larger foraminifera, but after the event that kills the dinosaur, we started to see foram is coming back to life, but they are much more smaller. So this is the typical foraminifera that we get in our Cenozoic timeline, but this is a much larger foraminifera during the dinosaur era. All right, so it's an interesting finding in Gubbio. So what else? Okay, so remember Cushman just now? The one that publishes the book, this is Cushman in the middle. So Joseph Cushman is the one that, put together everything in terms of taxonomy. He's the one that publishes the taxonomic book on foraminifera. And then he has two students under him. So this is Helen Tappan, and this is Alfred Leobich. So both of them are actually husband and wife. Right, so both of them are husband and wife. Both of them love foraminifera, and they publishes 2-volume of taxonomic books, which is an extension to the Cushman books previously. And this book has been used by us until today. There's no other recent, revised taxonomic book after Leobich and Tappan. And right now, if you are looking at foraminifera, if you want to look at the taxonomy of foraminifera, you still have to use the 1988, the year that I was born. All right, 1988 volume of taxonomic books on foraminifera. So as we see previously in 1920s, most of the foraminifera work has been focused on oil and gas exploration. But as we started to enter 1960s, most of this study shifted. They shift to understand modern foram. Why? Because if you look at modern foram, you can use your understanding in the modern foram to interpret past environmental condition. So it still is in earth science field. So by early 2000, somehow the study has shifted into foraminifera as biological indicator. But still, they are still in the earth science field, not yet as a biological marine field. So the, the shift is there. And if you ask me right now, what happened, we will talk later. The shift has moved on towards biology, which is they are trying to combine the geology and biological understanding to really find out how evolution happened, why do forams survive all this time, and what is the future for our ocean organism. All right, so let's talk about modern foram. This is a foraminifera. Yes, this is a big foraminifera. You seldom get them as big as this. I've seen a lot of much larger foraminifera. I think it was as big. I think it was bigger than 50, the size of 50 cent. If you're from Malaysia, you know our previously 50 cent, not the current 50 cent. It was quite big. I've seen that foram, but that particular specimen was collected from Okinawa, Japan. So this specimen, however, in my picture here is collected from Sabah. So we call this specimen the larger benthic foram. So if you are familiar with larger benthic foram, if you look at carbonate samples, you usually get them in thin section, but not as whole. I don't think you will get them as whole if you're looking at much older sample, but you will see them in thin section. All right, so you see them in their cross section of the foram. So what can we do with foraminifera and how can they become environmental indicators? So in the modern ocean, foraminifera has been recognized as one of the cheapest, mostly abundant, and statistically sound specimen in environmental study. Why are they cheap? Because you only have to collect sediments. You don't need any chemicals. You just need to collect sediments or maybe ethanol to preserve them. That's it. You don't have to, destroy the corals. You don't have to place traps in the ocean or things like that. You just need a sediment. All right. They are mostly abundant. You can find them in most sediment substrate in the marine. All right. So you don't have to go hunt for them. They are statistically sound because why? In 5-gram sediment, if you are talking about shaft environment, in 5-gram sediment, you can get easily more than 500 individual foraminifera. And if you're looking at deep sea, if it's an open water, you can get lots of planktonic foraminifera easily, more than 300 specimen of planktonic foram. All right, so they are very small in size, which means you don't need a very big carrier. You don't need a very big boat unless you're going to a deep sea, of course. You need a very minimum space to store them. And one more thing about them is that you see different species that likes to live at different place. I would say like if the environmental change, some species might stay, some species might go missing. So that is how foram is. So different set of environmental or ecological ecosystem, you have different foraminifera collection. So the assemblages or the collection of the species in the particular area will change according to the environmental changes. All right. Oops, sorry. All right, so this is how we apply foraminifera in our study. As I said, we are talking about the application since we know that foraminifera has a lot of, I would say, character that can tell us about the environment. Scientists started to use them to indicate what is a polluted environment. How is foraminifera living in polluted environment look like? So this study is actually in Italy. So if you can you can, think about Italy, especially Venice, you see a lot of ports, a lot of boating activities, and most of the time, if you have human and ocean, you know that there will be some pollution in the ocean. So what happened is that in one of the ports in Italy, they have a very high heavy metal pollution. So they detected, high heavy metal accumulation in that area. So they wanted to know what happened to foraminifera if the condition change and you have a lot of heavy metal pollution in the area. So this is what the foraminifera is supposed to look like if it's in a healthy environment. But instead, in Italy, when you have a lot of heavy metal pollution, they started to change in shape. You can see they change, they started to deform. So this one of the study that says that, oh, you can use forams as heavy metal pollution indicator. Once they started to change shapes, they are telling you that this is happening this is an area that have a very high metal pollution, and decision makers, managers should do something about it. So this is the application of heavy metal pollution. All right, next we are moving towards reef foraminifera. All right, as you see here, other than our heavy metal pollution, we can also use foraminifera as reef indicator. So how can we tell the health of coral reef from using foram? So we've carried out several studies in South China Sea. This is the study by my group, as foraminifera for foraminifera as reef bioindicator, and these are all the other studies that use benthic foraminifera as bioindicator in coral reef health. So how does it work? I think I would like to highlight this because this is much more on the biological side of work. People do not know that we can use foraminifera as indicator of coral reef. So corals are, if you know corals are actually animals. Yes, they are animals that require very warm, very clear water, an area with abundant sunlight. All right, because why? Because they have a symbiont or an algae that live inside them. So this algae will help make their food for the coral, and the coral will provide the algae a protection or a place or a home to live in. So coral live in colonies, but because most of them are very close to the coastlines, right? We usually find corals very close to the island or the coastline. They are subject to various threats. Naturally, they are subjected to climate change and also from human activities. So, well, why do we care about corals so much? Because they are the breeding ground for many fishes. They stabilize the ecosystem. They also act as a coastal protection and, protection from erosion for most of the islands. All right, so how can we relate foraminifera with coral? So if you look at larger benthic foraminifera, they have colors in them. You see this one are blues, and you see a slightly, I would say purple, it's actually red, and brownish foraminifera. So what happened is that in larger benthic foraminifera, they act like corals. They host algae, which means they create a home for the algae or their symbiont, and this algae will live in them. The algae will carry out photosynthesis, provide foraminifera some foods and things like that, and foram becoming the good host, which is like a house to them. And because of this, they require similar environment as corals. They require warm water, they require clear water, and they also require a lot of sunlight. So, which means you only get to see a lot of different larger benthic foraminifera in coral reef area. And as you move on to a deeper water, you will get less larger benthic foraminifera. And when the environment or the parameter at the coral reef area changes, maybe, there's a lot of tourists, there's a lot of organic matter, or somehow you get a lot of mud in that area, they will change very drastically, which means foram will change first. Foraminifera, the species will change first because they have short lifespan. Most of the larger benthic foram will live up to 2 months, 3 months. And as they change, their new generation will be the survival that can live in the changed environment. So if you have a lot of organic influence, you might not see all these beautiful bluish or purplish foraminifera anymore. All right, so this is a graphical, I would say a visual of what happened in coral reef area. You can see here, there's a lot of healthy coral, but as the environment change into somehow a lot of algae, the foraminifera will also change. You see, if you have a lot of healthy coral, you get this type of species. But as the environment start to change, where you have a little bit of coral, a little bit of algae, you started to see the environment change. So this study has been carried out in the Pacific I think it was in Sulawesi. I think it was in Sulawesi, Indonesia. And this is the foraminifera we gathered from Redang Island. All right, so similarly, we have the same species from Redang Island. You can see if the coral is healthy, we usually get this type of foraminifera, very roundish. But as the coral started to be covered by algae, you started to see the foram change in shape. So these are also from Redang Island. All right, and then scientists or ecologists started to think, why not we calculate the amount of species? As you can see, if you change, the environment changes, the number of dominant species that live in there also changes. So they started to come out with an index where we can calculate how much of the good foraminifera, which indicate good condition, and how much of the foraminifera that is stress tolerant. So it means they are not very sensitive. They can live in whatever environment there is. And we take all of this sum, we put it in an equation, and we get some numbers or values. So the values can tell us whether the environment is very conducive for reef growth, or was it a marginal environment, which is, so and so. I mean, if you get coral bleaching, coral might have 50-50 chances to recover. Or maybe the environment has changed into a very stressed environment, which means if you have coral bleaching or any drastic event from the climate change, such as, storm, things like that, coral might not be able to recover. So that is what foraminifera value or index can tell us. And we've been using this index in Tioman Island. So what happened is that if you are very familiar with Tioman, you know that on the east coast of Tioman is a very good or pristine diving area. Most of them are rocky shore, which is a lot of rocks. But on the west coast of Tioman Island, you get a lot of sand. So a lot of sand and a lot of residential and resort. All right, when you have a lot of residential areas and resort, obviously we know that we have a lot of pollution in the ocean. And what we see is that our foraminifera is telling us, the index, the one that I talked about, the index, is telling us that, well, this is a good area. If anything happen, the coral will, definitely recover. It's a good area for coral growth and things like that. But if you look at the foraminifera index that we collected from this area, especially E and F, it somehow is less than 4. So if it's less than 4, according to here, it is, it's entering the stress condition, or it's like a marginal condition for reef to grow. If anything happened, the reef might not survive anymore. So what do? Is that we also collected some sediment. So we see a lot of organic organic material accumulation in this area. And we see a lot of broken corals rubble in this area. And we also match our data with coral diversity data that has been collected by the other researcher. And we can see that, this is much more healthier coral in this area. This is okay. But as you go into this area, the coral, they started to have less diversity, only certain type of corals that live there. And if this, if we don't do anything about it, if we don't manage it well, most probably this coral will not even survive in the future. So that is how we apply our Foraminifera in looking at coral reef around Malaysia. So we've carried out a little bit of study in Redang. We're going to look at Perhentian as well and see what are the differences between these, marine park corridors here around the east coast of Malaysia. All right. So what else can you get from forums? So other than biological indicator for coral reef health, other than pollution indicator, Foraminifera has also been proven to be a very good sea level indicator, especially past sea level. So if you're in geoscience, if you're familiar with salt marshes, we don't have it in Malaysia. Most of our marginal area is covered by mangrove, but salt marshes forums in the subtropics or temperate regions, they have a very good Foraminifera to be a sea level indicator. Why? Because somehow different species distributed at different tide levels, and it is as good as plus minus 5 centimeter. That is how good you can predict past sea level. So through statistical calculation, you can get a very, very high resolution sea level reconstruction in the past. But unfortunately, in Malaysia, with mangrove, we have a lot of problems. One of it is because we have bioturbation. Bioturbations means you have a lot of animals digging around, introducing lots of oxygen, and then somehow Foraminifera will just dissolve and you can't get any good fossil samples. And we also have a problem with mangrove here because the elevation. So instead of going that way, in Malaysia, we have like elevation that goes up and down, up and down, up and down. So, well, not so good for sea level, high sea level indicator, but you can still somehow relate them with either sea level increase, rise, or falls, things like that. All right. Okay, then I think we are almost at the end. What else can we get from forum? Okay, so because I study a lot of modern foraminifera, my aim is to understand how foraminifera live, where do they live, and how they change in our surrounding water. Why do I want that? Because this data can be used for analog to the fossils record. Whatever you understand now in the modern forum, you can apply them to the fossils record and you can interpret or predict what happened in the fossils record. So right now, one of the project that I've been doing with my final year student is trying to help some of our friend in USM, which is currently studying the ancient Kedah civilization. If you are familiar with Bukit Choras, it was quite popular towards the end of last year, some discovery in Bukit Choras. So our friend in USM is telling us that, usually if you wanted to build a monument or some important buildings in the past, this civilization people will build it very close to the sea. Why? Because it's very convenient for them to reach with boats and things like that. So he thinks that our friends in Dr. Nasha thinks that Bukit Choras could be very close to the sea when it was first built. So he collected several core samples, a very short core samples, hand coral. So he collected some of it very close to Bukit Choras and some of it very close towards the ocean. So this is in Kedah, right? If you're not familiar with Bukit Choras, so this is very close to Sungai Muda, if I'm not mistaken. I have to check back. But anyway, he collected some of this sample and he wanted us to see if there's foraminifera or if there are any diatom or what can we do with the samples? Can we predict the sea level and whatnot? So what we found out that we found a lot of foraminifera in this sample and this sample. I am not sure about these two. I have to ask my student, but I certainly sure that this sample here and here has a lot of foraminifera, especially this species. So this species is usually a species that live from 5 meter to 10 meter water depth. So that is how the range of the sea level went once upon a time in this area. So could it be that Bukit Choras is, very close to the coastline? I think so, but we have to date the foraminifera first. So for dating, we usually send out to beta analytic for radiocarbon dating. When the dates come back, we can, provide an answer to our friend in USM whether, yes or no, the sea level is at this age and this level. So he just wanted to know whether somewhere around 1,000 to 2,000 before present, was it a coastal area or not? So we are trying to help him with that. You can do a lot more with forum actually. If you are very good with geochemistry, we have a lot of good machine and equipment right now. The technologies has advances to that extent that you can take one single foraminifera test, the small one, and you can get a reading from that single test. You don't no longer has to collect like 30 or 50 tests. You can use one test to determine what happened in the past. So the isotope measurement, this isotope, when foraminifera try to build their shell or their test, they will somehow store the isotope signal in the environment. So these isotope signals can tell us how the environment was. Was it warm? Was it very cold? So things like that. And we are using these advanced in technologies to really measure them. And once we have all the good modern analog, we can always apply them to our fossils record. And I would like to show you if you are working with biostratigraphy or if you're working with Miocene, marine forams. I think Miocene, not so much in our Malay basin, but if you're working with Sarawak basins or things like that, you can get a lot of planktonic forams. So, but not this particular species, but these are all the weird looking or nice looking to me planktonic forams that you can see here. This is the agglutinated foram. Remember the foraminifera that take sediment around it and glue it together to make its own home. So this is the agglutinated foram, and this is the typical coral reef forams that we found on the east coast of Peninsular Malaysia. So there is an endless possibility in foraminifera study. We might not need micropaleontologists on site for biostratigraphy, analysis as much as we did previously. I know geophysics study has advances a lot, but there is still an application in foraminifera to understand what had happened in the to the earth through the geological time. And we are currently trying to understand evolution of foraminifera through their DNA, a lot of metagenomics study. We are trying to do that. Most of the professor that is working on foraminifera previously they are like working with fossil forum right now has shift into working with genomic of the foraminifera. They are trying to combine geological understanding and biological understanding to predict the future. So I think that is it for me. Thank you so much for listening. So if you have questions, yep, I think that it's open for questioning.
All right, thanks a lot, Dr. Fatin. It was very interesting. And I think there is like also some lacks of maybe exposure in terms of like the usage of forum itself. And I think we are so used to like using forum for, like you said, oil and gas exploration. So perhaps if there's any questions from participants, you can either raise your hand or just turn on your microphone, or even leave in the chat if you would like to ask any questions to Dr. Fatin. Maybe I could start first. Where do you like? I don't know globally, and also perhaps in Malaysia, how much is like the advances of foraminifera studies, and also what have you yourself like? Is there a lot of people doing what you're doing, or also, or there's lack of that, and how would you like it to go on going forward?
All right, so around the world, there's a lot been going on. You see, well, I am trying to do both benthic forum and planktonic forum in Malaysia, but usually if it's out there internationally, they will just focus on either benthic or planktonic forum because the usage it goes back to your objective. What do you want to use them for? Do you want to interpret the paleo environment? Do you want to use them for evolution understanding, or do you want to use them for pollution related study? So I've been to several different places and look into several different of this field. The recent one is in Netherlands, which is much more, to my surprise, they study a lot on larger benthic forum and trying to understand the evolution. In terms of modern foraminifera study, within Southeast Asia, China has been very good at collecting samples and doing a lot of research, as well as deep sea study. China has been very good at that. But we started to see a lot of study from Indonesia, from Brunei as well, and well, we are trying to catch up from Malaysia because different places or different ocean in the world, they have different environment, especially our Malacca Strait that is, I think it's very unique. This is why our focus on Malacca Strait is a lot more, because if you understand, Malacca Strait is a little less saline compared to the rest of the world, and it connects the east and the west. So yeah, so in terms of the advanced technology, as I said before, they are looking at single cell forum. We don't have it here. I have to outsource my sample. I have to go to Taiwan or the US to get these samples analyzed. In terms of metagenomic study, I am not somebody that runs or play around with DNA sequences. So I have to get my colleague from biology to help me with it. And we hope that we can understand how forum adapt if climate change happen. Do they change their DNA? Do they change their protein and things like that? That is what we're trying to do here in Malaysia. I hope that, many more of geosciences researcher could join us or help us out with this research, because currently we are very limited in terms of foraminifera researcher in Malaysia. So yeah, we would like to form a very huge team if you're willing to.
Yeah, I think there's a lot of like networking, and also we need to highlight the interdisciplinary needs, because
Yes.
definitely it's not something that you can just remain in one field and then hoping for it to advance on its own.
True,
Okay,
very.
so I'll collect some questions from chat here. We have one from Muhammad Naim. Great talk, Dr. Fatin. The one in Kedah samples, is it the ammonia species? Thank you.
Oh yes, it is. It's the ammonia species. I think Naim has been doing a lot of work around Penang area, and I think he is also familiar with ammonia. Yes, it is an ammonia species. We found a lot of ammonia species. I am not sure about other species. I haven't looked at the sample very carefully. But my student has been reporting about ammonia species. So this ammonia species is a species that can tolerate a wide range of salinity, which means they can live in estuary or um, very close to coastal area, very close to coastal areas. And but their distribution in Malacca Strait is usually up to 10 or 15 meter water depth.
Okay. And there's another one here. TYX. May I ask that normally how many locations and samples you collect to ensure that it is a representative samples for that area?
All right. Because Foraminifera is very abundant, they seldom change in a large area. How do I mean large? Means if it's a shelf sea, you will usually get the similar species, but the amount of the species may differ according to water depth. So if you are interested in looking at an area, you need to know what are your bathymetry is, which is very important. You need to know what your adjective is. Then you can start to collect samples. So if you are looking at modern analog, let's say you have a core samples and you are looking at modern analogs, we usually recommend that you collect more than 40 samples of surface samples to represent the modern environment. The more sample you get, the better. As you can think of core sample, it usually change the environment sometimes change from marginal to fluvial, and sometimes you go back to like a shelf area and things like that. So more samples, the better. But if you can get 40, then it usually is good enough.
Yeah, I guess it's always in any field work we would want more samples.
Yes.
Okay. Hassan Muhammad said that hope this finding can contribute to Kedah civilization and perhaps other tamadun. I guess foraminifera is as well indicator, like you said, because it can be used for like throughout the entire the age of the earth. So definitely it can contribute to a lot more things. What about, do you think on is there any other groups that currently are doing what you are doing with the applications of foraminifera, modern foraminifera, other groups in Malaysia that you know of?
For modern, I'm not so sure, but I know a lot of people doing on the older fossil forams in UMS. I know some lecturers in UMK, in UKM, if I'm not mistaken. They've been doing a lot on Miocene, a lot of different age, which if you ask me, I'm not very familiar, not that familiar with, because my study focus on Holocene and modern. So right now, Dr. Zalia is still in UMT, so we have two of us in UMT. She's the only one that I know working on forums, and we recently pulled our colleague from biology to join us to do a lot on the DNA sequencing. That's it. I think that is all on that I know. I'm not sure whether there are other researchers, but if you are there, please reach out to us. We would love to collaborate with you. All right, so I think Dr. Khaira has a little bit problem with the connection. So if there is any more question, I can let me read the chat as well. Regarding the civilization of Kedah tour, I am also very excited to see whether we can use the Foraminifera to match the age with the Bukit Choras. And if that finding is, telling us something, then hopefully it can get published and hopefully we can get a lot more knowledge on how Kedah tour started, which is I'm very excited to see. And there's also a few more spots of ancient civilization along our west coast. And if I can get the opportunity to, join the studies, we'll definitely update somewhere. Hopefully Foraminifera can at least shed a light on how the sea level works, because we know that the sea level changes in different patterns at different time of the Holocene, but it will be definitely good to see what happened towards the late Holocene period. All right, so Dr. Khaira is here back.
Hello, sorry, my internet is not stable. So I think it's already 11 and it's okay for me to wrap up as well. The talk, thank you so much for giving us a very fruitful thoughts, I think, and also hope that could encourage more people to look into different things and different research in geoscience.